Morning. We're coming to you from the Cochin Institute, and we are very pleased to be here with Dr. Roberto Malone and a team of young researchers who are working in his laboratory along with the laboratory in Sylvain U. We have three different young scientists to present their research. But first, a word from Dr. Malone.
Thank you, Monica. Welcome to this virtual visit to our INSERT Cochin Institute lab in Paris, which is associated with the diabetology clinic of the Cochin Hospital on the same site. So our focus, or the focus of our research, is type 1 diabetes, and those are very exciting times in the field, and we are sharing this excitement and contributing to some of these advancements with many different colleagues around the world. The research is made by people, researchers, clinicians, people living with type 1 diabetes, and I think that our lab makes no exception, and we have a number of talented scientists that work every day on this, and you will hear some glimpses of their research in the next presentation. So just to put a bit things into the big picture, we are here now starting our journey in the clinic, where our questions often start from our everyday medical care experience, where our questions often transit because we have a lot of work ongoing with samples taken from patients that generously contribute to this research, and where our research sometimes achieves its final objective as the most promising treatments are tested in clinical trials. And the type 1 diabetes field, as you may know, is going through revolution because we now know the benefits of early management of type 1 diabetes. We have teplizumab as the first disease-modifying drug, and we have a number of things that will be upcoming to pursue these new options of treatment. But this is still not enough, and what we are trying to prepare in the lab is the improvements that are needed for the next steps. First, autoantibodies are the mainstay of type 1 diabetes screening, but have some limitations, so we are trying to develop some additional biomarkers that may stratify disease risk and also give some prognostic signal about the efficacy of therapies under trial. Second, we know that these new immunotherapies are starting to have some impact on the natural history of the disease, but we need to develop the next generation of treatment that will be both immunomodulators and beta cell protective agents, and also to find ways on how to prioritize these treatments based on preclinical data and how to efficiently move into clinical trials. And third, we still know very little about the etiologic factors and the pathogenic mechanism of type 1 diabetes, and this understanding is also critical to improve our risk stratification and to identify new therapeutic targets. And with this, enjoy your visit. Thank you.
Thank you so much. Really appreciate you taking the time for this intro. And our first speaker today is Fatoumata Samassa, but we will go with Fatou. She is here as a postdoctoral researcher in the laboratory of Roberto Malone and Sylvain Yu in Paris, France. Her work is centered in type 1 diabetes with the goal of unraveling the mechanisms by which the immune system mistakenly attacks the body's own insulin producing beta cells in the pancreas, and she aims at identifying new biomarkers predictive of disease onset by focusing on CD8 and T lymphocytes and their specific peptides presented by pancreatic beta cells. She recently had a very nice bioarchives preprint, and we're very interested to hear more about her work in the laboratory. Thanks for joining us.
Thank you very much for this introduction, and I want to thank you for giving me the opportunity to present our work, which focuses on beta cell antigen recognized by autoreactive CD8 cells. So I'll start with a short introduction. Type 1 diabetes is an autoimmune disease characterized by the destruction of pancreatic beta cells by autoreactive CD8 cells. During a long pre-symptomatic phase, there is a progressive reduction of the beta cell mass several years before clinical diagnosis, and the only available predictive biomarkers of the disease are autoantibodies. But what makes these beta cells visible to the immune system? Islet reactive CD8 cells recognize via their T-cell receptor a beta cell peptide that is presented by HLA class 1 molecules at the surface of the beta cells. This triggers the cytotoxic activity of the CD8 cell, leading to the beta cell death. As you can see here, there are several HLA class 1 molecules, each of them being very polymorphic in the human population, and actually each of them leads to the recruitment of a different CD8 cell repertoire because they represent different set of peptides. One of the histopathological hallmarks of type 1 diabetes is the increase of the expression of this HLA class 1 molecule, which is triggered by the inflammatory milieu dominated by type 1 and type 2 interferons. But what about these beta cell peptides? A few years ago, our lab and others have shown that islet reactive CD8 cells isolated from PBMCs displayed a similar frequency and a largely naive-like phenotype in both type 1 diabetic individuals and healthy control. This suggests the existence of benign autoimmunity existing in all individuals. So we wonder, have we been looking at the right antigens? Indeed, the known antigens were identified as targets of human autoantibodies or as being targeted in the non-mouse. However, no evidence of natural processing or presentation by human beta cells was then shown. Also, all these autoimmune T-cell responses previously studied are HLA-A-restricted. So what about the peptide presented by other HLA class 1 molecules? We wanted to use an unbiased approach to identify presented islet antigen. And in a previous paper, ECN90 beta cell line were cultured overnight with inflammatory cytokines prior to cell lysis and peptide HLA class 1 immunopurification. Then, following a peptide isolation, the peptides were identified using mass spectrometry and bioinformatic tools. As expected, inflammatory cytokines induce an increased expression of HLA class 1 molecule at the surface of the beta cell, which is paralleled by the increased number of peptides identified in this inflammatory condition. Also, this study provided the first catalog of HLA class 1 peptidome of human beta cells with the identification of several peptide and source proteins, among them several coming from the secretory granules. However, this study came with some limitations, one of them being the study of neo-antigens, which was overall limited, and the second of them being the presentation of specific peptides induced by inflammation, which was relatively understudied too. So we aimed at characterizing more extensively the modulation of the beta cell immunopeptidome, first by studying post-translationally modified peptide in one hand, and in another hand, the peptides that are presented in the inflammatory condition. And we focus on interferon alpha stimulated beta cell. Why interferon alpha? Because it's the earliest cytokine signature in at-risk individuals, which appear even prior to autoantibodies. So first of all, the number of peptides identified in the interferon alpha stimulated condition was twice higher than the one in the basal condition. And this is parallel with the increased expression of HLA class 1 at the surface of the beta cell. As already reported, we identified several proteins, but in this study, we identified more peptides and new source proteins among them a lot of granule proteins, including chromogranule B, secretogranule 2, and secretogranule 3. Now, regarding neo-antigens, and in particular post-translationally modified peptide, here you can see that 32% of the one identified were specific to the interferon alpha stimulated condition. And among them, I want to draw your attention to this one, the S-glutathione, which is a huge modification adding 3 amino acids to a cysteine. And this is actually the major redox system of the cell, so it may reflect the status of oxidative stress of the beta cell. Now, if you focus on the HLA class 1 restriction, we described a significant enrichment in HLA-B binders as compared to other restriction in interferon alpha stimulated condition. And actually, when we look at the abundance of this peptide, we see again the increase for the HLA class 1 peptides in the interferon alpha stimulated condition, and this seems to be driven by HLA-B binders. This is concomitant with a preferential increase of the expression of HLA-B molecule at the surface of the beta cells in the inflammatory condition, so interferon alpha stimulated one. And as you can see, this is also the case for interferon beta and interferon gamma stimulated beta cells, as you can see here. And actually, when we dig into the literature, we realized that this observation went completely unnoticed in several papers, including this one, in which they performed single-cell RNA sequencing of beta cells in type 1 diabetic and non-diabetic islets. And you see that the HLA upregulation followed the exact same hierarchy than the one I just described. Next, we wonder, does the histopathological hallmark of HLA class 1 hyperexpression in the islets of type 1 diabetic patients also preferentially involve HLA-B? So we collaborated with Sarah Richardson, and we had access to a pancreatic section that were stained with anti-HLA class 1 antibody or anti-HLA-A or anti-HLA-B. So we had section from type 1 diabetic individuals with insulin-containing islets, but also non-diabetic individual. Here is the quantification of the expression of the HLA molecule at the surface. And as expected, we observed an increased expression of HLA class 1 in type 1 diabetic sections as compared with non-diabetic section. But again, HLA-B seems to be hyperexpressed to a greater extent than HLA-A. And last, we asked whether this preferential HLA-B regulation in type 1 diabetic slices also translated into recognition of HLA-B-restricted peptide by islet infiltrating CD8 T cells. To this end, we actually tested 29 HLA-B-restricted peptides, and we assessed the HLA-B-restricted response in CD8 T cell line raised from islet infiltrates of HLA-B40 type 1 diabetic donors. So these 29 HLA-B peptides were divided into 4 pools, and here is the results for 3 donors. And you can see that for each of these pools, there was a positive interferon gamma response for all the donors that were tested. We also started the deconvolution of these pools, and here is the example of donor 2, where we deconvoluted pool 1 and pool 2, and you can appreciate which of the peptides were responsible for the positive response we are seeing in the above graphs. To conclude, I've shown you that interferon alpha stimulation increases the antigenic visibility of beta cells, and this leads to an enriched, diversified epitope presentation. We identified new epitope, and including post-translationally modified neo-antigen and interferon alpha skewed epitope presentation towards HLA-B ligands. Altogether, this suggests that the HLA-A-restricted CD8 T cells studied to date might have missed a relevant repertoire of HLA-B-restricted T cells. Here is our working model. Just to briefly conclude, in a healthy islet, we think that there is a low expression of HLA-A molecule and almost no expression of HLA-B, while in type 1 diabetic islets, when there is insulitis, there is a preferential upregulation of HLA-B, leading to the recruitment of HLA-B-restricted CD8 T cells and increased beta cell targeting as compared with the HLA-A-restricted CD8 T cells. This leads to several questions for the next step of this project. are these cells better biomarkers of the disease? And for that, we will assess the frequency and phenotype of circulating HLA-B-restricted CD8 T cells in type 1 diabetes patients and in healthy donors. And also, are these cells more pathogenic? So for that, we will assess their killing efficiency as compared to HLA-A-restricted CD8 T cells in interferon-stimulated conditions. And with that, I want to thank all the people involved in this huge project, the funding, and you for your attention.
Thank you very much. That was a beautiful presentation. We're going to open it up to questions. Those who joined later, please drop your questions in the chat. We can field them. Also, I had a couple questions for you. Why do you think HLA-B is more unregulated than HLA-A in interferon alpha stimulated conditions?
Yeah, this is a really good question that we thought about. And actually, by looking at the promoter region of the gene encoding for HLA-B, we realized that there were two interferon responsive elements, while it's only one in the promoter of the gene coding for HLA-A. This might explain why HLA-B is more regulated than HLA-A in interferon alpha stimulated condition.
Only in beta cells?
That's a good question. I will have to check that.
Yeah.
This is a
No worries, it's just curious.
Yeah.
How do you plan to assess the higher pathogenicity of HLA-B restricted CD8 T cells?
Yeah, for that, we actually have a technology that is running in the lab, and you will hear about it in the short later. And this is actually briefly a transductant that we are generating. These transducts are transduced with TCR, recognizing either HLA-A or HLA-B peptide. And basically, we will have these two transductants, and we will put them in contact with interferon alpha stimulated beta cells and assess the killing efficiency of these cells in inflammatory conditions.
That sounds really exciting. We'll look forward to seeing what's happening in laboratory downstairs or upstairs, wherever we're going. Next. But thank you again. That too is great. And next next coming up, Widad Nefida. Yes. Okay, it's joining her talk is going to be insulin allergy as a model condition to test tolerogenic therapies prior to T1D prevention studies. She's a PhD candidate in immunology at the Institute Cochin in Paris, driven by a deep commitment to advancing therapies for autoimmune disorders and allergies. Her research explores insulin allergy as a model condition to test tolerogenic therapies prior to T1D prevention studies. And with a master's degree in immunopathology from the Sorbonne, she is passionate about bridging the gap between laboratory discoveries and clinical applications. Throughout her academic journey, she's completed several internships ranging from identifying biomarkers for autoimmune conditions to testing innovative therapies for these disorders, and she's eager to contribute to real-world healthcare solutions. She's keen on pursuing further research opportunities where she can apply her expertise in immunology and therapy development. And just as a sidebar, which I didn't mention, she's an MD, PhD candidate. So thank you so much for joining us.
Well, thank you very much for your introduction, and thank you for giving me the chance to talk about this rare condition, which is insulin allergy, and the way we use it for type 1D prevention. So in our lab, we aim to develop a safe immunotherapy to prevent type 1 diabetes in its early phases, so possibly before any autoimmune reaction occurs. Meaning that this therapy is designed for very young children. So to keep the safety profile very high, we use an antigen-specific therapy with a non-invasive route of administration. So we developed the PPI-FC, FC fusion protein composed from the preproinsulin, which is the inactive form of insulin, insulin being the main antigen in type 1 diabetes. Linked by a linker to an FC fragment of a human IgG1, which extends its half-life and take advantage from the neonatal FC receptor pathway that is physiologically used by antibodies to cross mucosal epithelial barriers. So our construction can use the same pathway and cross the mucosal barriers to increase its bioavailability. So our strategy is using the PPI-FC intranasally to prevent type 1 diabetes in a non-invasive way. So we use the intranasal route, which is perfect because it's non-invasive. It's linked to the tolerogenic environment associated with mucosal tissues. And also the FcRn are expressed in the nasal epithelium. However, early type 1 diabetes prevention trials are very challenging because they need large screening, long follow-up, and also we need a very robust safety profile since we're dealing with young children. So it's high risk, high cost, and this is why we thought about a de-risking strategy, namely insulin allergy. So insulin allergy shares the same antigen as with the type 1 diabetes, which is insulin. So this offers a rare opportunity to apply the same therapy to both an autoimmune disease and an allergic reaction. So insulin allergy is a rare but serious skin reaction, complication of insulin therapy that occurs in diabetic patients. It causes different symptoms such as wells, flares, and can also lead to life-threatening anaphylaxis. So it can be immediate or delayed allergic reaction, and measurement of specific IgEs and skin testing are the common ways to diagnose insulin allergy. To date, there is no treatment for insulin allergy. Patients are commonly redirected to insulin pumps, which is not always adequate for their needs. So testing the tolerogenic effect of PPI-FC in insulin allergy is so advantageous because the follow-up is very short, the safety profile is flexible because we are dealing with adult patients, so it's low risk, low cost investment for clinical trials. First, we had to develop a mouse model of insulin allergy. So we used an NOD insulin 2KO mice. So it's a model that lacks one copy of insulin gene, making mice susceptible to developing severe autoimmune type 1D. So these mice were sensitized twice a week for 3 weeks with insulin and allen, and the control group was sensitized with PBS and allen. 1 week after the last sensitization, we challenged mice in the left ear with insulin and the right ear with PBS. So the PBS ear is an internal control for each mouse. 3 minutes after, we inject Evans flu intravenously. So Evans flu dye technique is a commonly used technique in allergy to see the extravasation, meaning the local allergic reaction, as shown here in the picture. We have the ear challenged with insulin of a mouse sensitized with insulin that appears blue. 10 minutes after, we collect the ears to quantify the optical density of the Evans flu and other organs that are important for our analysis. So for our strategy, we desensitize mice with 2 therapeutical windows, one which is an early desensitization. We start the desensitization before the last sensitization, so with intranasal PPI-FC, and another therapeutical window, which is a late desensitization, meaning we start the desensitization after the last sensitization. There is a lot of sensitization in this sentence, sorry about it. So as you can see here in the pictures and also in this graph, with our sensitization protocol, we increase the Evans flu in the ears, meaning that we have a local allergic reaction. And with our treatment with both therapeutical windows, we decrease this local allergic reaction. And this goes with the total IgE that is decreased with our PPI-FC intranasally. This is also the case of the histamine release when we do a mast cell degranulation assay, meaning that our PPI-FC managed to lower the local and systemic allergic reaction. We then analyze the immune response related to allergy and its desensitization. So allergic reactions are often shown as an imbalance between the Th2 and Th1 response, which are 2 populations that are CD4 T cells. And when we have an allergic reaction, we have an increase in Th2 response, while the Th1 response remains low. So we set up an in vitro assay to quantify IL-4, which is the signature of Th2 response, and interferon gamma, which is the signature of Th1 response. So we stimulate polyclonally these CD4 T cells or in a specific manner to insulin. And in both cases, we have the increase of the Th2 response, as you can see here with the IL-4 secretion. And we managed to lower this Th2 response with our treatment in both therapeutical windows. Meanwhile, the Th1 or the interferon gamma secretion remains unchanged or slightly increases with our PPIFC intranasal. So with this PPIFC, we restore this balance between the Th1 and Th2 response. Then we considered the possible mechanism behind the efficacy of intranasal PPIFC in reducing insulin allergy and focus on IL-10, which is a cytokine commonly associated with nasal tolerance. And we use the same setup as I showed you before. And indeed, we have the increase of IL-10 production in this population, in this T-cell population. And interestingly, we have an increase in IL-10 cytokine within the B-cell population also. So we are inducing with the nasal PPIFC a T-cell and a B-cell response. And to demonstrate that the IL-10 is the key mechanism behind the efficacy of intranasal PPIFC, we blocked the IL-10 using an anti-IL-10 receptor while treating mice with the intranasal PPIFC. So as shown here in the pictures and also in this graph, blocking IL-10 inhibits the effect of PPIFC. So we have this increase in the optical density in the ears. We have the same levels of IgE as an allergic mice with the blocking of IL-10, and it's the same for the mast cell degranulation assay. Importantly, this effect is also inhibited when we look at the Th2 response. And also this effect is inhibited, of course, when we look at the IL-10 response in the T and B cell population. So as a conclusion, we managed to develop a very robust experimental model for insulin allergy, and we proved the concept of using an intranasal Fc fusion protein to have a systemic and also a local effect. So we identify IL-10 as the key mechanism of this treatment. And of course, now we are trying to analyze this Treg and Breg response and also looking more into this insulin-specific B cells to see if they play a role in this whole story. Also, we want to investigate the long-term effects of insulin PPIFC with distant challenge or several challenges. And of course, we are treating our mice or our NOD mice with intranasal PPIFC to prevent type 1 diabetes. And I hope next time I will show you some interesting results. I just want to emphasize the importance of this project because this strategy could be applied to other types of allergies. Also, we can maybe try to start a clinical trial here in the center at Cochin Diabetology Department because it's the reference center in insulin allergy. And of course, I think that the non-invasive way of administrating the PPIFC is very important and very innovative for type 1 prevention. So thank you for your attention. And I want to thank the lab, the collaborators, and also for facilities and funding. So thank you so much.
Great, thank you so much. This is a huge effort here and really innovative, you know, in terms of trying to address that pediatric population, especially. I loved how you were thinking about the de-risking. I have a couple of questions. We're opening it for questions. And I have a few questions. Have you tried other non-invasive routes of administration?
Yes, we tried the oral route as a non-invasive way of administering the PPIFC in another project for preventing type 1D with the same protein, which is the PPIFC, and we had a partial prevention. But the problem is the destruction of this PPIFC with the gastrointestinal degradation. So this is why we thought about an easy way to prevent this degradation. So it's the non-invasive way. So it's an intranasal way. It's easier than making other compartments around the PPIFC to make it not be degraded with the oral route.
Yeah, and you also mentioned that this strategy might be successful with other kinds of allergies. Have you tested this with any other allergies?
Yes, actually, we tested this with the ovalbumin allergy, which is a way stronger allergy, and indeed we had very good results about decreasing the allergic reaction locally and also systematically. So we are very happy about these results, and we think we can make it also for other types of allergies.
Wider reach. Do you have any preliminary results on the PPIFC and T1D prevention?
So now we started this new project of prevention, about the type 1D prevention. So we have seen some encouraging results. We have a delay in the type 1D appearance in mice, and now we are doing more experiments to look more into this.
Nice. And then last but not least, do you have any data on regulatory populations responsible for the PPIFC therapeutic effect?
Actually, this is very preliminary results, but we saw a slight increase in the Treg population when we look in spectral cytometry. So we will look more in details about that, and
Fantastic. Well, it's really interesting. We can't wait to hear what you have next to present. Maybe what are you thinking? When's your next sort of big release of information?
I think it's the IDS Congress.
IDS in
So
November.
I don't know yet if I will have an oral presentation or a poster. But please, if you are interested in insulin allergy or type 1D prevention, please come to my presentation or poster. I would be happy to answer. And also maybe at this time we'll have more information about the type 1D prevention. So
Sounds great.
Thank
Thank
you.
you. Widad Nefida, PhD candidate. Thank you very much. And I kind of pre-introduced already Clementine, and she is an MD PhD candidate. Just a little bit about her. She's a young physician specializing in diabetology, practicing at the Cochin Hospital in Paris. Since the start of her medical studies, she's witnessed the field of diabetology transform at a rapid pace thanks to advances in research. Always fascinated by the pathophysiological mechanisms underlying diseases that she manages, she took the leap in 2020 and enrolled in a master's program in immunology at the Sorbonne to contribute to advancements in her field. And she's currently working on a PhD thesis in the team of Roberto Malone and Sylvain Yu at the Cochin Institute, focusing on the mechanisms that could explain the heterogeneous progression profiles of patients with type 1 diabetes. So today her work is divided between her PhD thesis, clinical research, and clinical practice. Very busy schedule. I believe that my knowledge in immunology makes me a better physician, and my clinical practice provides a critical perspective on the work I do in the lab. By bridging the gap between the bench and patients, I hope to be a valuable asset to her team, she said. And yes, I have heard that, you're really bridging that gap very successfully and well. So at the end of your talk, I'll ask you a few more questions about how you managed to do that.
Okay, so thank you for this nice introduction, and I'm very glad to introduce you our T-cell being asset to model the fundamental vulnerability and resistance of the cells in vitro, and also to assess the crosstalk between T-cells and beta cells. So first, why do we need such a model? So as you all know, type 1D is an autoimmune disease where eventually the pancreatic beta cells are destroyed by CD8 T cells. So beta cells present their endogenous peptide to the CD8 T cell via a molecular complex called HLA class 1. CD8 T cells recognize those peptide by their T cell receptor or TCR and release into immune synapse cytotoxic granules, leading to beta cell death. So beta cells do have some weapons to defend themselves, as for example, they express at their surface PD-L1, which after linkage to PD-1 at the surface of the CD8 T cell will lead to T cell activation. but there are more and more evidence that type 1 is also a disease of the beta cell itself, with the beta cells being too visible or too vulnerable to CD8 T cells also. And now that thanks to exogenous insulin injection, type 1 patients are no longer doomed to die within months after onset, there is more and more interest in finding a cure to the disease, either by acting on targeting the CD8 T cells or by decreasing beta cell vulnerability, or of course, both. So nowadays, a lot of possible molecular targets and drug candidates have been identified. But to confirm that these candidates are able to protect the beta cells from CD8 T cells also, we need a human reproducible high throughput model to test and to study the mechanism of these drugs. So for such a model, we need at least a reproducible and unlimited source of beta cells. And this we already have, as we have several immortalized beta cell lines, or we can also use iPS islet-like cells. We also need an unlimited and reproducible source of CD autoreactive CD8 T cells directed against peptide, against beta cells peptide. And this is a bit more tricky to obtain. And we also need a high throughput and reproducible results. So first, how do we generate our unlimited and reproducible source of CD8 T cell directed against peptide of beta cells? To identify the CD8 T cells in the blood of donors, we use a technique which is called the tetramer stain. So the tetramer is composed by a molecule of streptavidin linked to a fluorophore. And this streptavidin molecule has 4 sites of linkage for a molecule of HLA class 1. And those HLA class 1 are preloaded with our peptide of interest. So in our case, peptide presented naturally by the beta cells. We put these tetramers with PPM6 donor, and they will link to the CD8 T cells, which have a TCR specific for this peptide. And after, thanks to the fluorophore linked to the streptavidin molecule, we are able to sort the CD8 T cells specific for our peptide. After the sorting, we perform a sequencing of the TCR, and we re-express this TCR in an lentiviral virus. And so we can perform a lentiviral transduction of primary CD8 T cells, and then we obtain this primary CD8 T cells expressing all the same TCR of interest directed against the beta cell peptide. And once we have performed the step 1, 2, 3, and half 4, we just have to repeat the step 5 and 6 to generate as many CD8 T cells as we need. So now we have our beta cells. We can add on top CD8 T cells, autoreactive CD8 T cells with TCR directed against beta cells peptide. And now we can add in the well a different concentration of glucose or cytokine to mimic type 1D pathophysiology. And we can also add different drugs and molecules to modulate the interaction between the beta cells and the T cells. And that leads us to the readout. How do we assess the killing of the beta cells by the CD8 T cells? So I want to present you today the 3 main techniques that I working on. So the first is the real-time imaging. So in the real-time imaging, you put your beta cells, you put your T cells on the top, and you add a reagent. It's a kind of a live-dead reagent that will stain the beta cells which are dead. And so the machine will measure the area or the number of dead beta cells over time. So for this example, I use the beta cell exposed to interferon alpha. As we know, interferon alpha is a major cytokine, it's a type 20 pathophysiology. And we treat also these beta cells with a drug that we call today compound A. And A is an anti-interferon alpha drug. And so you have beta cells exposed to interferon alpha, treated or not with compound A, and we put on top CD8 T cells treated or not also with compound A. And what we expect is that compound A will decrease beta cells lysis by the CD8 T cells. And indeed, this is what we obtained. But surprisingly, the main effect of compound A seems to be directly on the T cells, as you can see on the red curve. So this technique is really interesting because it's a real time. You can study multiple time points, and you will need a very low number of cells to perform. Second, I will talk to you about the plate reader, but it's a fluorimeter. It's a fluorimeter. So the example is exactly the same. It's beta cells exposed to interferon alpha and treated or not with compound A, and cultured for some hours with T cell cluster not with A. And here we observe exactly the same result. Compound A is able to protect partially the beta cells against CD8 T cells assault, and the effect of A seems to be mostly acting on CD8 T cells, as we can see on the red curve again. And more even surprisingly, it's is also able to protect beta cells from the CD8 T cell assault in absence of interferon alpha treatment. So this is a technique that is very simple and very fast for the experimenter. You need a low number of cells, and there is a limited variability. And last but not least, the flow cytometry. This is our gold standard technique, but it's a bit more time consuming to perform. And so in this example, beta cells are exposed to interferon alpha and treated or not with a TYK2 inhibitor. And so TYK2 is a molecule that is part of the interferon alpha pathway. And so we expect that the inhibition of TYK2 will decrease this beta cell death. And indeed, when we expose beta cells to interferon alpha but treated them with the TYK2 inhibitor, we see a decrease in the lysis, but we do not go back to the level of lysis of beta cell not exposed to interferon alpha. And thanks to flow cytometry, where, as you know, you can look at several markers, we could show that this is mainly because TYK2 inhibitor decrease the level of HLA class 1 expression, but also decrease the level of PD-L1 expression by the beta cells. So the flow cytometry is really useful because you can look at multiple markers and so explore the mechanism of action. And you have very limited variability, but it's self-consuming and time-consuming. So to conclude, the real-time imaging helps you to select time points. The fluorimeter allows to screen multiple conditions at once, and the flow cytometry allows a mechanistic study of the chosen time points and conditions. So to have the whole picture, you have these reproducible models with a limited source of beta cells, on the top of which you have your limited and reproducible source of CD8 T cells. You have in your well everything you want, glucose, cytokine, and your drugs. And so you can do now as many conditions as you want. And you have your different readouts. So with the real-time imaging and the plate reader, you can define the time points and the conditions that are really interesting to study. And after, with the flow cytometry, you can really go in the depth of the mechanism of the condition you have chosen.
Okay. Very nice, thank you very much. Really interesting, set of protocols and an approach. I think we have a couple of questions for you, and we're opening it up for questions to the audience. Here's a couple of questions from us. You do not perform a knockdown of the endogenous TCR. Doesn't that lead to background noise with non-specific killing?
At the beginning, we thought that maybe we should perform a Cas9 knockdown of the endogenous TCR, but it would have made the process heavier. And in the end, what we see is that when you transduce your CD8 T cells with the exogenous TCR, you have a very important expression of this TCR at the surface. So you have a lot of TCR at the surface. And some of our collaborators also think that maybe the endogenous TCR is internalized in the T cells. So maybe at the surface you have a lot of exogenous TCR, and maybe you don't have that much endogenous TCR. But in the end, what we did is that we have a TCR directed against a peptide that are not usually expressed by beta cells. So if you want an interaction between this CD8 T cell expressing this TCR and the beta cell, you have to preload the beta cells with this peptide. And if you preload the beta cells with this peptide, you will have a killing. But when we did not preload the beta cell, we didn't observe any killing. So we think that the noise is not that very important.
Good, thank you. It's good. That's good due diligence there. I wondered when will you release the name of compound A?
In fact, there is it's point A here, but in fact we have a lot of work between A, B, C. And we will see maybe at the end of my thesis, I will have to.
Fantastic, yeah. No, I just was kind of curious. That was very curious. And then, sort of just apologies again to Widad, which I slotted her in the end, getting her doing both MD PhD. She's just, doing her PhD is enough. But you do have a rare profile. You're doing both MD PhD. And we were told that you grasp both sides of this equation very well. So what's your secrets? How are you balancing your clinical and research interests, and how do they complement each other? Kind of interesting, we have a presentation coming up with the DiabDocs program that's based in the US. It's an NIH-funded program that's sort of headed by Linda D'Ameglio and Maz at Stanford, and they really try to recruit MD PhDs to the T1D space. So I just wanted to see, kind of get a feeling from you or a sense from you how you're managing this.
How I'm managing this, I'm not sure that I'm managing it perfectly, for sure, because it's a lot of work and it's a lot of jumping to one task to another. And yes, so I think I'm managing this thanks to the kindness of Roberto, who is accustomed to such a timetable, can a bit. But I think that having the both sides make me a better physician and a better scientist. Because, for example, as a physician now, I'm questioning myself a lot about the etiology of the diabetes in my patient. Is this really the type 1D? Is this really type 2D? What can I do better? What can I do more for this patient? And on the lab side, I think that brings an interesting point of view, because sometimes I can say, okay, this is really interesting, but why the patient should care? What this discovery will bring to a medical field?
Reality check.
Yes.
Yeah. Yeah, I do think that's what we've heard as well from the DiabDocs. They had their first cohort last year, and now we're showcasing their second cohort. It's that it does inform both sides of the equation, and it's it gives you a unique perspective. So that's fantastic. All right, then I think everyone did a beautiful job presenting in a very clear and detailed way what is actually going on in each of your research programs. And I really thank you for taking the time and speaking to us and to our greater audience. We do record these sessions and they are housed on our video catalog, video library for future viewing. So thank you again, everyone.